Hello, good afternoon, everyone. Good evening, good morning. The webinar is joined by people from different countries. You are welcome to the fourth webinar of HoliCare on lamp primer design. We have three guest speakers. Dr. Amman Russom, Professor Amman Russom, and Dr. Mohammed Bakheit, and Dr. Friedrike Hans. This is our fourth webinar. If you would like to tap into our previous webinars, you can join HoliCare Academy. A web link will be shared. My name is Edgar Napoleon. I work for the Swedish program at ICT Involving Regions, which is one of the consortium members in this project. I will allow me to mention that we are going to have surveys posted live during this webinar. So if you see a pop-up or a poll, just answer the questions and close. This will be done twice at the beginning and somewhere in the middle. Right now, let me invite in Professor Amman Russom. Amman Russom is a chemical engineer by training and a professor in nanobiotechnology at Royal Institute of Technology, also known as KTH, here in Sweden. He heads the nanobiotechnology division at KTH. He obtained his PhD from the same institution, KTH, before his postdoc fellowship at Harvard Medical School. Amman is the head of the HoliCare Consortium, and he coordinates the project overall. He is going to tell us about HoliCare project, and he'll be followed by our guest speakers. So over to you, Amman.
Thank you very much, Edgar, for this great opportunity to be here with you and give you introduction about the HoliCare project. And then following myself, we will have a webinar about lamp primer design. Anyway, let's get going. Just to give you a very brief introduction, the background, and then about our consortium, and then I will have just one final word before I give the floor back to you, Edgar. So what you're looking at here is a picture. This was a review from 2014. What it's showing basically is that antimicrobial resistance is going to be surpassing cancer when it comes to mortality rate per year. So which really highlights that we are into a big challenge dealing with infectious diseases and most specifically antimicrobial resistance. If you look at this picture here, this is what has been reported to WHO as hazard between 2001 and 2013. Really, infections are dominant. And another thing you can see is there's really high concentrations at resource limiting settings. So there is a big need addressing this. Another recent publication, I would urge you to read it, is that about half of the world population do not have access to proper diagnostics. And around 19% of the population in resource limiting settings have access to simple diagnostic tests. So which means there is a big unmet need when it comes into diagnostic needs at resource limiting settings. And if you look at this, one of the key mentioning in this review was that the level of primary care in so-called diagnostics, the so-called last mile, particularly affects poor rural and marginalized communities globally. So all in all, access to diagnostics in resource limiting settings is a major driver to the high mortality rates. So if you look at specifically at respiratory tract infections, this is a global view of what has been the, let's say, highest ranking in terms of mortality global. So cardiovascular cancer are on top. But if you look at it specific to the burden of resource-limiting settings, you see that lower tract infection and diarrhea are among the top burden. So this has been really the focus of HoliCare, is looking at respiratory diseases. And the HoliCare is a consortium that has been active since September 2022. And we're looking at specifically disease-related, poverty-related diseases, and more specifically respiratory diseases in Sub-Saharan Africa. So the question really is, how do you improve disease management diagnostics in rural area and or technologically underdeveloped geographical locations? So we have been really fortunate enough to build this consortium where we're looking at technology advances, but also combined it with capacity building implementation in order to address this critical bottleneck. And in doing that, we've been focusing on a 4-pillar approach. In one end, we're developing next generation diagnostics. And then in the other part, we're looking at capacity building, human capacity building, adoption, implementation. So it's a 4-pillar approach. I'm really fortunate and happy to say that this capacity building has been one of the most important pillars in order for us to bridge the gap between diagnostic innovation and implementation. So when we talk about point-of-care diagnostics, this is the type of test that could be done at anytime, anywhere, by anyone. So we're looking into speed, portability, quality, convenience of use, etc. And when used at resource-limiting settings, we normally talk about and criteria we call assured. Assured stands for affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable. And very recently, this was updated with 2 more letters. This is reassured, which reflects the need to have real-time connectivity, ease of specimen collection. So why is this important? Basically, what I'm showing here is it's a pyramid where tertiary care is at the highest level, and then you have the primary care is at the lowest level. And what we are looking at is that can we have diagnostics or point-of-care diagnostics that can help us to take what normally used to be at the secondary and tertiary care all the way to the primary care. So which means can we develop technologies that can allow us to do things that you traditionally would only be able to do it in the secondary and tertiary care. It's very, very briefly. And so the HoliCare consortium as such is then trying to address this using this 4-pillar parallel methods that approaches that I have been talking about. And who are we? We are 14 partners across Europe and Sub-Saharan Africa. I'm particularly super happy to mention that we have 4 partners from Sub-Saharan Africa, from Ethiopia, Senegal, and Uganda, part of this core team trying to address this bottleneck. We've been funded by the European Commission. This is one of 7 parallel projects that has been funded in this program, Innovative Approaches to Enhance Poverty-Related Diseases, Research in Sub-Saharan Africa. And with that, I would like to mention my final word. This is that alone we are excellent, together we are brilliant. So I'm really happy to kick off the webinar number 4, which is going to be about LAMP primer design. And with that, I'll give back the words to you, Edgar. Thank you for listening.
Thank you, Professor Amman. For those of you who have just joined, we've had an introduction about the HoliCare project. And now we will move on to the main theme of the webinar, which is LAMP Prima Design. We have two guest speakers. But if you want to know more about the project, you can visit the project page and see how we are progressing, bringing on board these solutions. So allow me now to introduce our first speaker, Dr. Mohammed Bakheit. He is the head of development at Research and Development Department, Mast Diagnostica in Germany, where he has been working since 2014. His education background is veterinary medicine. He obtained his bachelor's and master's degree from University of Khartoum, and he has also a doctorate degree from University of Free University of Germany, Berlin. He did his postdoc, worked in diagnostics, specifically in serological and molecular diagnostics, and he worked also at the National Reference Center for protozoan diseases at Obihoro University of Agriculture and Veterinary Medicine. His focus has been on diagnostics, and before joining MAST, he participated in development of diagnostics for tick-borne protozoan diseases using LAMP. The next guest is Friedrike Hans, who studied biochemistry and molecular biology at Christian Albrechts University in Kiel, then obtained her PhD in molecular neuroscience. She also has done postdoc in molecular mechanisms for neurodegenerative diseases. He's also working at MAST since 2003. Dr. Mohammed and Friedrike, the floor is yours. You are most welcome.
Thank you. Thank you very much, Edgar. And I also say good morning, good afternoon to everybody. And thank you that you are here. And we are happy now to start with our joint presentation with Friedrike on the basics of LAMP primer design and so forth. So the presentation itself will be divided into three pieces, three parts. The first part would be a small introduction on the LAMP technology, very brief, and then there will be the most important part, and it is about to select sequences and how to define a target. For that, we had already also previously, the previous seminar done by Dr. Hamid Ismail is a very big help in this part. And there will be a third part, which is a demonstration on how to perform primer design using this online tool called Prime Explorer. I'll be talking a little bit about it then in the third part. So to start with the introduction part, loop-mediated isothermal amplification, which we also know as LAMP. The word is coming from the first letter, these red letters, and the AMP is one amplification method, which is for the isothermal amplification of DNA and RNA. It's a molecular diagnostic method, and it was firstly published in 2000 by Notomi et al., who was working and is still working for the company Aikin Chemical in Japan. There is over the years increasing interest in the method, and I've just checked this today. As of today, there are exactly 4,820 publications. But for the molecular diagnostics, the method is also very interesting, and it has very big advantages. One of them is its suitability for many applications, including point-of-care diagnostics, as Amman has just mentioned. We are involving this method in our HoliCare project, and the panels which are delivered in this project will be working with the LAMP method. It's another advantage that it's very affordable, it's relatively cheap reagents. The polymerase itself is also not that expensive. And most importantly, there is no need for expensive equipment to perform the LAMP. So it's more suitable for resource-poor settings and outside developed laboratories. Another very important characteristic is that it's tolerance to inhibitors, which is actually helping also devising DNA preparation methods, which are very simple. That's to say, not very pure, but enough to run the sample. And this is very important as time to results also becoming very important. And would like to mention that we are involving this also in HoliCare project. So what are the basic requirements in order to perform LAMP? Just like almost every other DNA or RNA amplification method, you would need to prepare the reaction environment by the so-called reaction mix or reaction buffer, which is usually salts. There is a buffering system there. There are also the DNTPs and so forth. There is also a DNA polymerase, but this one has a very important characteristic that enables the LAMP reaction, and it's the fact that the polymerase used here should have strand displacement property. That's to say, it's able to nick out a DNA strand while building a new one, as I will demonstrate shortly after. The secret behind LAMP and its ability to work lies actually in these specially designed primer sets, and they enable then the isothermal amplification in an automated way under a fixed temperature. Now I would like to just briefly describe these primers, as it is actually the subject of this.
Sorry, Mohammed, I can't hear you from here. I'm not sure if it is the case with the rest. Yeah, I see one post in the chat. Could you check your sound, please? No? As our presenters fix their sound, for those of you who have just joined, you can find some of the important links in the chat room, and if you have questions, we will have a Q&A session, so you can post them in the chat, and we will read them to our presenters.
Excuse me, do you hear us now?
Yes. Thank you.
Okay. Great. Thanks a lot. Yes. So as I was mentioning, the requirements for the LAMP reaction, and these are the reaction buffer, the DNA polymerase, and the primers. And the primers are composed of two sets: the outer primer sets and then the inner primer sets. The outer primer sets are marked in yellow color, and we have the inner primer sets. Each of them is composed of two parts. The first part, which we call the F2 primer, it's a little bit to the outer part, and then the inner part, which is the F1C primer, which is to the inner part. And both parts together compose the so-called either forward inner primer or backward inner primer. There are additional optional primers, which are called the loop primer. And these are basically to accelerate the reaction. They enhance the reaction in terms of time, and they are positioned in the middle between the two parts of the FIP or the two parts of the BIP primer. So we have on this side, in the reverse strand, we have the loop F primer, and on the other side, we have the loop B primer on the forward strand. Very shortly on the principle of the LAMP amplification, And as mentioned previously, the lamp amplification works. Yes, and this is the most complicated slide in the whole presentation, but I will try to manage it now because it just summarizes how the lamp works. Usually the reaction is initiated. We have here, do you see my mouse? We have here, for example, a target DNA strand. And the reaction is usually initiated by one of the inner primers. As I was previously mentioning, we have this fifth primer, for example, and it's composed of the two parts. So there is the annealing part, which anneals to its corresponding region here. And this will be then taken by the polymerase, the extension, until the end of the strand. But then thereafter, we have an outer primer, and it has also an annealing position in the same target strand. So there will be the ability to initiate also another extension with the BST polymerase. And as I mentioned, that enzyme is able now to nick out the previously built strand and to build a new strand. As I'm now showing here, this is now the extension of the F3 primer and nicking out of the previously built strand with the FIP prime. Now this nicked out strand has a really very nice characteristic, and just look at the end here, you will see this F1 part and the complementary part to the F1. So these ones will then anneal together to form the so-called loop shape here. They just anneal here together, and I have this structure. The same thing is also happening from the other side, driven by the backward primers. Again, the B prime in this case initiates a reaction, followed by a B3 primer, an outer primer, which nicks out the previous strand. And this nicked out strand is now having this particular shape here, which we call a double dumbbell structure. And as soon as we have this structure, then it's actually the starting structure for the automated amplification. If we look closely at this structure, we find annealing places for many primers. So we have here, for example, the F2C position. It's where an FIP primer can anneal. We have here B2, where BIP primer can anneal. And if you include these optional primers, which we call the loop primer, that means you have two additional annealing positions for two additional primer, and a fifth place where extension can take place is this one free 3 end here. So altogether you have 5 places to initiate extension and DNA building. And this process now goes without the need to do anything under isothermal condition. It's very powerful, and in 30 minutes you can have something like 0.5 microgram DNA amplified in a reaction tube. So the method is very fast, and there are several ways to detect the umbilicon that's coming with this method. You can do it also even with the naked eye, but most likely then fluorescence measurement or turbidity measurement and other ways. Today I see also electrochemical measurement and so forth. So for those who are interested to learn more about the principle of the lamp, we place these 2 video recommendations. They will be also shared in the publication. And that's actually the short introduction which I wanted to give. Now we forward you to Friedrike to continue with the sequence and how do we select sequences for the lamp.
Yeah.
This one.
Thank you, Mohammed. Yeah, so in the next about 20 minutes, I will talk to you about how to select suitable candidate sequences for the lamp primer design. And therefore, I will introduce to you the NCBI BLAST tool and the MSA Viewer and sequence alignment tools. So first, here you can see an overview about characteristics that a suitable sequence for lamp primer design should meet. First, it should be pathogen specific or stage specific gene. This means a developmental stage, like in parasites, where you want to detect a parasite in a certain developmental stage. Second, the sequence should be highly conserved within the species of your interest, in case you want to develop an assay for a certain species, or for a group of pathogens, in case you want to detect like Aspergillus species, or another group of pathogens. Third, the sequence should be highly stable. This means it should not be prone to genetic mutations. You often have this in viruses, where like genes that encode for proteins of the envelope or so are very prone to mutation. And then fourth, it is nice if the sequence has a high copy number, but it's not a must. And then fifth, the GC content should lie between 40 and 65%. But it's also okay if it's a little bit lower or a little bit above, but usually the primer design and the LAMP assay works best if the GC content is in this range. So how can you find out a pathogen-specific gene that you can use for a primer design? So the identification, you can just go on PubMed, the literature research base, and search for molecular diagnostic assays that were published before for your species of interest, and search which target genes were used there. It doesn't matter if it's a PCR method, another isothermal amplification method, or the LAMP assay. And then there are also other databases like information banks that contain information on a certain topic. For example, there's an information base for Mycobacterium tuberculosis, and here we have listed the LAMP Primer Bank. This is a collection of published LAMP assays and target genes for respiratory pathogens. So this fits to the HoliCare project. And then, of course, there are bioinformatic analyses of whole genomes, which can be performed to identify new sequences which are highly conserved in your species of interest, but which are not present in other species. Okay, so then you have to make sure that the target gene that you have identified in, for example, the literature, is really highly conserved within your species. And therefore, you can do a blast of a sequence, of the gene. And with that, you can also find out how stable this gene is, and also get information on the copy number. And for BLAST, this means that you compare your sequence of interest with a database that contains hundreds and thousands of other sequences, also of the same gene. And there you can then compare and see if there are mutations or gaps or insertions. And from that, you get information how conserved this sequence is. So first, you have to get a sequence. Therefore, you can just go on NCBI's, and there are several databases. You can search in the nucleotide database, in the gene or in the genome database, and search for your species of interest and the target gene. Here I have made an example for Mycoplasma pneumoniae. That is one of the targets, one of the pathogens from the HoliCare Academy. And the 16S gene, which is a typical high copy number gene in prokaryotes. And there's a sub-database, the reference sequence database, which contains annotated and curated sequences. And this means that these sequences have a very high quality, that they can be trusted, that they don't have gaps or wrongly sequenced parts. And there you have the possibility to download the FASTA sequence. Now I'd like to switch to the browser.
This one. Ja.
Ja. Nee, wir sind noch nicht im Browser. Okay, it does not work.
Which one?
I want the viewers cannot see the browser now. They just still see the presentation. I'm sorry again, we have another technical problem.
If you update your sharing, so if you go to your share tray, then you can update and pick the browser instead of the presentation.
Yeah.
There you go.
Okay. Okay, good. Now we see the correct screen. Okay. So here we have a FASTA sequence of the 16S gene from Mycoplasma pneumoniae. As we've learned already in the Bioinformatics webinar, this is composed of two parts. Here we have the header line with some information about the sequence, and here the accession number. And down here is the nucleotide sequence. And we copy this sequence and go to the BLAST page. So BLAST stands for Basic Local Alignment Search Tool, and it is a tool to find regions of similarity between biological sequences. And since we have a nucleotide sequence here, we go to the nucleotide BLAST. And here you have to enter information of your sequence and of the parameters with which you want to run the BLAST. So here we enter our 16S sequence. Here you have the option to give the BLAST a name. I recommend that if you run several BLASTs at the same time. So I just call this. Then you have to choose the database against which you want to run the BLAST. There you have here several options. We recommend to use the RefSeq genome database because there the sequences are of very high quality. But for example, for eukaryotes, there are only one sequence per organism listed in this database, and there it is better to use the nucleotide collection database. And there are also many more databases and also for RNAs, and you can find more information if you click here. Then you have to enter here the species in which you want to do the BLAST. So we want to do it in Mycoplasma pneumoniae. And then you have to search the program. And for us, there are two options. If you want to run a BLAST in the same species, then you use the highly similar sequences program or the MegaBLAST. But if you want to do a cross-species BLAST, so there are more differences, then you should choose the more dissimilar sequences or discontiguous MegaBLAST program. And then finally, before hitting the BLAST button, you can change some algorithm parameters. And here I would suggest to change the maximum target sequence. So the default value is 100, but I would set it to 5,000 so that you get the most hits as possible. And then you start the BLAST. This can take just a few seconds, but also very long. And therefore we have already prepared the BLAST here. And here you can see the results. In the upper part, there's a lot of information about the BLAST. For example, here you have the query ID, and query means this is the sequence that you're running the BLAST with. And yeah, here we have the query length of 1,487 base pairs. But of more interest to us are the results, which are here in the lower part. We have a table with many data. And you can see here that in total we have 288 sequences as hits. And of all the data, only 3 columns are of interest to us. This is here, which gives an information about how much of the query sequence is covered by the hit. So if it's 100%, the hit covers the whole length of the query. Then we have the E-value, which describes the number of hits one can expect to see by chance. So the lower this number is, the more significant the value is. And here we see that all the hits, they have an E-value of 0. This means they were not found by chance, but these are really significant hits. And the third column that is of interest to us is the percentage identity column, and it gives an information about how identical the hit sequence is to our query sequence, and 100% means there are no mutations. So, and I deselect here, and then we scroll down to see if there are other hits which have less than 100% identity or coverage. And when we go down here, we can see that there are here hits which have an identity of 99.93%. So there are some mutations in these sequences. And we want to analyze them further. So I just select some of them randomly. Here I select some more. And we just go further down in the list, because the further down you go, the less similar these sequences are to the query sequence. Here we select some more random sequences for the next analysis step. And here at the bottom of the list, we also have some hits with 99.87% identity. So we choose some of those. And here at the bottom, we also have two sequences with a query coverage of 35 and 25%. So they are not of to us. We are only interested in sequences where the query coverage is high. And now there's a nice tool with which we can see an alignment of all the sequences that we have selected now. And this is here, the multiple sequence alignment viewer. If you click here, and your window will open. And here we will see an alignment of all the sequences that I've just selected. Here at the top is the query sequence, so the sequence with which we run the BLAST. And here are all the hits that I've selected. And you can see here that there's a red line. This means there's a mutation. And if you want to know more about this mutation, you can zoom in here. And we have to move this field to the side here. And you can see that in the query sequence, we have an adenine base at this position. But the mutation here, it is a guanine, and in all of the sequences, this means this is a conserved mutation. And we zoom out again. And you can also see that here in the lower sequences, there's a conserved mutation. And this is a single mutation in one sequence, but you don't find it in all of the sequences. But you also see that the remaining part of the sequence here between the mutations, and also here in the end part of the sequence, it's conserved in all of the different 16S sequences from different genomes. So this sequence, the 16S, is really highly conserved in Mycoplasma pneumoniae, and therefore is suitable for further analysis so that we can find out if we can use it for a specific primer design. Now I have to go back to the presentation screen.
Just try if that's going to work. This presentation.
Yeah, no, it works. Thank you. I have prepared screenshots of everything that I presented to you in the presentation. And yeah, now we have tested or found out that the 16S is conserved in the Mycoplasma pneumoniae species. This is the first step. But then you also want to know if the sequence, if there are significant differences in other species that are closely and further related to your species of interest. So in this case, for example, in the Mycoplasma species. And therefore you should repeat the BLAST with the 16S sequence, but in the Mycoplasma group. And it's optional. You can also do further BLASTs in the family, domain, or kingdom to find out how big are the differences to other species of this sequence. Here I have prepared a screenshot of an MSA viewer of a gene from another pathogen from the HoliCare panel, which is Haemophilus influenzae. This species has a very heterogenic genome. And here you can see very well that in all the sequences, there's at some positions, or at many positions, conserved mutations spread over the whole sequence. And here it is not possible to find one part of a sequence which is conserved in all of these different sequences. But still you can use this for primer design because you can do an alignment of representative sequences from all of these sequences and use this for a specific LAMP primer design. And to do such an alignment, there are several tools. We like to use the Cluster X tool. Mohammed is going to introduce you to this tool a little bit later. And then there's a web-based tool, the multi-line tool. And as you can see here, it gives a very good visibility of the variations between sequences. So here there are mutations between these two sequences and also here, and they are coded with a different color. So they're really easy visible. And we like to use it, for example, for documentation of our LAMP primer design. There are also other tools that you can use for preparing alignments, which we have shown here. Yeah, and then finally, when you have performed all the BLAST analyses, you have to choose which LAMP primer design mode you want to use. And yeah, as a help for the decision, I've prepared this decision tree. So you have to first ask yourself, is the target sequence highly conserved within your species? If you say yes, like for the 16S Mycoplasma pneumonia sequence, then you have to answer the question, is the target sequence significantly different in other closely related species? If here the answer is again yes, then you can use one single sequence for the primer design. But if the answer here is no, there are not very big differences of the sequence to other closely related species, then you can do a specific LAMP primer design where you use one sequence from your species of interest and further sequences from the other species which show a high identity. But if you answer this first question with no, like for Haemophilus influenzae, as you have seen, then you can do a common LAMP primer design with an alignment of representative sequences from this species. Yeah, and with that, I'd like to give back the hand to Mohammed for an introduction on LAMP primer design.
Yes, so and we have decided here to just give a brief introduction. Actually, there's a lot of information there on LAMP primer design using Primer Explorer software. We will talk a little bit on the requirements of Primer Explorer on the sequences and sequence alignments and few lines on the basic primer design, but also advanced primer design. Why do we choose Primer Explorer? It's one of the first tools which are available for everyone online and free. And if you visit the website of Primer Explorer, you will find until this moment there are two versions of the software offered, this version 4 and version 5, by clicking on this link here. And version 5 is a little bit more advanced, so maybe you can use this one. They keep updating also the tool. And in this moment, there are also the manuals being published. If you navigate to this web page here, you will find a very big manual on the use of Primer Explorer, which I recommend to read. You can download these 3 PDF files then. For the purpose of the demonstration, we have made screenshots of Primer Explorer with example of the operations that we are going to show here. But first, let's talk on the sequence requirements of Prime Explorer. So how do you prepare your sequence to load to Prime Explorer? You have identified your target, as Friedrike has shown. Now, put into your mind that the program does only accept a maximum of 2,000 base pairs. That's one strict requirement of Prime Explorer. And that your sequence should only contain the 4 classical letters, so no degenerate sequences. It only accepts A, G, C, T. And the software also accepts single sequences, but also sequence alignment files, which are called .aln files. You can upload this or this, and you can then start with your sequences. The software thinks about primer design. It has fixed parameters that are always in the consideration. This should then generate the best primers for the lamp. And the factors which are considered, I bring now back again the picture of with the primer set for the lamp. So it's always considered that the melting temperature of this primer, of the inner primer, are around 65 degrees, in the range of 64 to 66 degrees for the F1C and B1C parts of the FIB and BIP primer, but also for the loop F and loop B primer. For the outer primer, the F3 and the B3, but also the F2 part of the FIP and the B2 part of the BIP, the temperature can be a little bit lower, so around 60%. Considering that your lamp incubation temperature is going to be something between 60 and 67 degrees then later. Another factor that the Primer Explorer considers in its design is how the 3 ends of your primer should be, and what is called the stability of the primer ends. These important ends could be 3 ends. So that's for the F3, F2, for the B3, B2, but also for the loop primer. But for the most inner primer here, most important are the 5 ends. So you have always to check the 5 ends of your F1C and your B1C parts of the FIP and the BIP primer. And the requirement of the program is that it calculates the free energy, and it should be -4 kilocalorie or less. So if it's -5, it's going to be even better stability than -4, and -6 is even much more stable. I need to mention at this stage that the design of the loop primer, these 2 optional primer, always in a separate step in Prime Explorer. So you first need to have your 4 main primer, and if you are happy with your primer set, then you can proceed to design loop primer set as I will show. Now the third other key factors that are considered by Prime Explorer in the primer design are also the length of the target, of the lamp target. The distance between the beginning of the F2 until the end of the B2, that's the lamp amplification product. This one is usually set to be in length between 120 to 160 bases. If it's more than that, then the lamp may be a little bit slow. You try to make it as short as you can. And another important distance is also from the beginning of the F2 to the beginning of the F1 parts of the FIP, or here the BIP primer, from the beginning of the B2 to the beginning of the B1C. And this region is important, especially if you come then later to design a loop primer, because they are exactly inserted somewhere here between the 2 regions. So you make sure that there is enough space here for that. The software considers that the GC content of the designed primer always between 40 to 65%, and that the secondary structures and hairpins, there is also a set up default DG threshold value for the dimer check. and it is, I think, minus 2.5, something like this. If it is higher, it's fine, it's much better. That means the probability that your primers interact with one another or form hairpins is lesser. So minus 2 is better than minus 2.5. Now, for the purpose of demonstration, I would like to talk a little bit on basic primer design, but take an example of using a single sequence for the primer design. And with basic, we mean that you leave everything to the program to do. That would mean that this is the first start page of the program. There is a browser button here. When you click on it, it will open a window where you can direct it to your sequence. And if you remember, the sequence should fulfill the criteria that we talked about previously. And next, you can click on primer design here at the bottom. This will lead to the fact that your sequence will be uploaded and you will see it in this window here. You can make sure that that's a single TXT file will be displayed here, then Spiralis fumigatus point TXT. And to the right side of this window, there are additional functions. A little bit down there, you have this design option. And when you are using a single sequence, please always use the default option for your design. Now you don't need to do anything other than to press on generate. When you do that, the software will start automatically searching the sequence, taking into consideration the factors which we mentioned, and it comes back to you with a summary of how many primer sets were generated. As you can see here, 5 primer sets were generated. They are also displayed here. And by clicking on the display button, you can display these primers, as I will show in the next screen. But below this button, there is a detail setting button. And this one is the one which you are going to click if you want to modify anything. In other words, if you want to do a little bit advanced or to have control yourself on the primer design, which I will show later. So now we press on display, and as soon as you do that, the software will jump into this screen. And as you see, there is your sequence displayed, and also these 5 primer sets are displayed in the sequence. The software automatically gives them ID numbers, like in this case, and they are actually being completely selected by the software itself. You can, for example, for the purpose of demonstration, you can click on number 1, for example, and you can press on confirm, and now you will see that primer set being displayed. There are a lot of information being also displayed with the primer set. Here in this green window here, you see the forward outer primer, the backward outer primer, but then you have also the whole FIP primer with its 2 parts and the BIP primer with its both parts. And these parts are also individually displayed down here. Now you can see also the 3-end stability of your prime. If you remember, the value which was the default value was -4. You can see now we have much better because they are less than -4 here for the F3, for the B3, and for the F2 and for the B2. As we mentioned, for the F1C and the B1C, more important are the 5-end stability value. That's 1 here and that's 1 here. The default one here is -3. And the primer is fulfilling the requirements for Primer Explorer. What you can do is to press on primer information, and this will generate a file which you can keep in your computer. This file contains information on the sequence that has been used. You will need later to add loop primer to this primer set. Now let's go to a design of loop primer. And as I mentioned, you need that primer information file. Again, you start with the same window, the same screen, pressing browse, but instead of loading a sequence, this time you load that file being generated from Primer Explorer, and you press on Primer Design. The view is now this time a little bit different. You have your sequence there. This time the Primer information file name is displayed. But in your sequence, as you can see, you also see where the exact position of that primer set that you designed, and now you are attempting to add the loop primer to it. Just to remind, the loop primer are inserted between the F2 part and the F1 part of the FIP, but also between the B2 part and the B1 part of the BIP here. When you press on generate, now the software will start searching for suitable primers, and it comes back to you with a number here. And in this particular example, 11 primer pieces were generated. You can press on display, and again, you will see your sequence. You will see the sequence of the primer set, the original, and now you see that there is some suggested loop primer from Primer Explorer. But I see that there are only suggested loop F primer. There are this in this region, the software couldn't find a loop B primer. So if you are happy with your primer set, you can now decide whether to go with the original primer set without loop primer, or just to include this one, or to manually design another one here, or to use that advanced setting button to modify or to ease a little bit the conditions that the software is going to consider in order to see if you can have, if you can force him to design a loop the primer for you. That was the choice which we did. And as soon as you press on this, it was basic, it was here advanced designing, then it will open a window with additional things that you can modify. One of these things is the length of the primer, the melting temperature. You can also modify the GC rate, the stability. You can ease these values a little bit. And we have studied that region, and in fact, the GC rate of that place was a little bit less than this 40. So if you would just reduce this a little bit to 38 or something like that, if you would accept 1 degree less here with your melting temperature, and you can attempt again design, you see that this time there is a suggested loop B primer for you. So now you have altogether 15 primer sets. I mean loop primer sets. And you want to choose one of them in order to add to your original primer set here. You base now your choice on two things: the 3-end stability of your loop primers, that's a very important factor to consider, but also the DG dimer of your primers, the ability that they form hairpins with themselves or some sort of dimers between themselves or with the rest of your primers. As I mentioned before, the higher this value, the better or less is the probability. So it's clear that my set is not going to be one of the first 3 ones, which all have minus 3.4. It will be one of the below ones here. Now I know that all my loop B primer are having exactly the same end, so they should have also exactly the same affinity value. What I will have different will be the 3-end of my loop F primer. And that's where I take some examples of different loop F primer ends and study them. Now I can view them here. And what you can see is that this pair here, LF, has a 3N stability of minus 5.56, and it's actually the best one. The other ones, all of them are having much lesser than that. As mentioned before, the loop B should be exactly the same one. So this is now going to be your primer set, and it has also the lower dimer value. By pressing on save, and I mentioned this here, but it's always there somewhere, a save button, which is going to help you to export your data to an Excel file as it is in this particular case. So that's a demonstration of a basic primer design where you don't need to modify anything other than these few things which we did for the loop B primer. But now let's do a little bit more with these modifications, but also taking into consideration in our example now the use of the sequence alignment file. And the use of sequence alignment file enables us of many other different design strategies. You can design specific primer for specific species of a genus. You can also design a common primer set. And a sequence alignment file actually stores the information on sequence alignment, which contains many sequences, and present this information to Primer Explorer. So like, for example, in the display, you can see where there is an insertion in the sequence, you can see this small dot here. Where there is a deletion, for example, you can see it's always displayed as small letters. In between these two small letters are a deletion. Or if there is a mutation, it's going to be in minus here. And where you see a star, that means there is no difference here between the sequences of your alignment. To prepare such an alignment, now here there is a small trick. Primer Explorer doesn't accept any alignment prepared using any tool. This one I would recommend because it looks like it's Primer Explorer friendly. You can use this cluster X. We have placed the link here where you can obtain it. And here is Friedrike has prepared for us a little bit of sequences that are all in FASTA format and in a text file. And this is what cluster accept to upload. You can use the file button here and you can direct it to that text file and you can load now your sequences here. This is not yet a sequence alignment because you have here in the alignment bottom here, you have additional options. And remember about these options, especially the sequence number. I don't know, it should always be on for the sequences to be uploaded successfully to Primer Explorer later. The output order is also important. So you can keep this as the input order like that, or you can leave it for the program to decide which sequence should be at the top. So in case that you are going to design specific primer set of a specific genus from an alignment which contains relevant or relatives of that genus, and you want to drive your primer to pick only primer which should amplify that species but not the others, you have to ensure that that sequence is at the top, because it will be then used as a bucket skeleton for the alignment, and the information on the other member sequences in the alignments will be then inserted to that sequence, like what I've shown here previously. So you make sure that the major sequence here is from the organism that you are going to design specific primer set for him. If you are going to use the sequence alignment for common primer design, then you don't need that thing. Now you prepared your sequences, you chose these options, you now press on do complete alignment, and the software is going now to generate several files. And Among these files, you will find a file with extension .aln, and that's the one which contains the sequences which you need now to upload to Prime Explorer. Again, back to the main screen of Prime Explorer, browse, you direct him to the ALN file, and you can press on Primer Design. Now let's do that directly on the screen. Thanks, Friedrike, you prepared now some files. Now here are Aspergillus sequence alignment files, and now this is the file which I want to upload. It's this ALN file. Now it already displayed here, and I press on Primer Design. Now the ALN file, as you can see here, previously when we used a single file, it was TXT file, now has been successfully uploaded to the window. And as you can see in the alignment, there are also that information I talked about. Like here, for example, you can see that there is a minus there, which means that this region contained a mutation. Or here, for example, in this window now, you have several other possibilities, but what I mentioned before that for the purpose of using an alignment, you want to do either a common or specific primer design, and this you can select here from the design option. Now we are going to demonstrate a common primer set, which is supposed to amplify all the organisms in that alignment. If I now press on generate, that means I'm not modifying anything other than showing the software to search for a common primer. That would mean please avoid wherever there are problems with the sequence, wherever there are mutations, wherever there are insertions or whatever, these regions should not be incorporated to any free end of any important primer. The best would be to avoid them. And the software has already found 5 primer sets for us, which are fulfilling a common primer requirements. That's again basic primer design. But now let's try to do something else. To go here to the detail settings. Pressing the detail settings will now give me additional options here, which I'm going to go through now. But let me just demonstrate one thing. Just by pressing that detail button, the software understands now that you want to have the control on the design. That would mean that it will not even suggest to you any primers now like it did before. That's to say, when I just press on generate, not modify anything, this time I have 602 primer sets. Previously, I used to have only 5. That means these are the 5 ones which have been selected by Prime Explorer. Now it doesn't want to do this to me. It will say, okay, this sequence enables 602 common primer, and it's for you to choose what you want. Now let's see what are we going to choose. If you go down here to the characteristics, to these parameter conditions, you will see that Prime Explorer considers 3 different modes for primer design, depending on the GC content of your primer, of your sequence. When you have normal, you would see that the GC rate, which is considered here, is between 40 and 65. If you choose 80 rich, for example, you see that it has now switched to a little bit 80 rich mode, a little bit longer primers, a little bit lesser melting temperature, or you can choose also the GC rich mode, and you see that the primers are now a little bit shorter and the melting temperature is a little bit higher. And because these modes are overlapping, so you can actually try designing primers with all these 3 different modes.
It's not always possible for every sequence.
Exactly.
Yeah, but you can always try.
Yes. Now I revert back to the normal mode and go to describe this region again, which is the DG threshold, which is again calculating the DG threshold for the stability that we mentioned before. Now these are the default values. Now if you want to make your primer search more stringent, you can reduce these values. I go here to -4, for example, and for this one to -4.5, for example, -4.5. And you can even make this one, this for the dimer check, you need to go a little bit higher, so maybe -2 instead of 2.5. And for the distances, if you remember previously, we talked about 120 to 160. That's going to be the best value. And now you can press on generate. Now you see, because you made the conditions a little bit more stringent, it has now filtered out a lot of primer sets, and now we are just left with 14 primer sets. Well, you can make the conditions much more difficult until you end up with only one primer set here. That's possible, but, and pressing on display, you see that with much more stringent settings, you are actually limiting also the software to only one small region of your sequence. That's in this particular case, this region starting from here to, I don't know, 1,400 something. And now all these 14 primer are actually very, very similar to one another. They are very similar to one another. Yeah, that's so, and but that's already a very good number to start choosing one primary representative from this. And now we come back to that character which we talked about before, which is the stability of the 3 ends, and also the dimer check. That's the dimer check value. In this particular example, all of them are having exactly the same dimer check, so that's not going to be a problem. But let's see the stability of the 3 ends. Now you see here, this one is the same. The 3 end of this one, no, it's not the same. I have 2 variants. I have this variant which is ending with G, and I have another variant ending in GG. Let's see further. Here I have exactly the same 5 end for the F1 prime, and exactly the same 5 end for the B1 prime. On the other side here, I have exactly the same 3 end for the F2, for the B3, and for the B2 prime. That would mean I have now, just to check which one of these two is giving me the best stability, choose both of them here from the primer ID and go to that bottom, which we say we shown before, confirm. And now I see that I have 2 primer sets. And I can just jump directly to the F2 primer and to the 3 end of that F2, because that's where the difference was. And now I can check the stability of that F2. For the first one, it was -7.37. And for the second one, it is -7.97. So the second one is even much better than the first one. So that one is now, this one is now going to be my primer of choice. And I can just press on primer information file and save this information file, which is now also again saving the information of the sequence alignment as well as the primer sequence, and I can use this one now to go again and to design loop primer as I've demonstrated before. So these are very few tips on using sequence alignment and on using these advanced primer design tools. It's okay. So we also prepared in this slide additional information, which I covered now in the demonstration. So they will be in the presentation. The importance different windows and screens and how to deal with them is also present in the presentation. And with that, we would like to say thank you very much for your attention. We are now ready to take your questions or anything.
Thank you. Thank you, Dr. Mohammed and Dr. Friedrike, for the interesting presentation. This was very interesting for me. We have about 10 questions.
Oh, wow.
And if I may, let me start with the first one. What are the strategies or techniques do you employ to mitigate the risk of non-specific amplification in LAMP assays, particularly when dealing with complex samples?
Okay. Yes, the non-specific amplification is a problem of a non-well-designed LAMP assay. At the same time, it's a problem of reagent. So this problem can be tackled in several ways, actually, or several factors are to be considered. One of the reasons for false positive amplification is what we try to avoid here in the primer design, and it is the fact that some primers could be interacting with one another and initiating some sort of reaction in the background. And this is actually one of the first criteria which we choose in our testing when we come to testing new primer set, that there is very, very minimum to no background amplification. That's to say that the primer will keep always a very low background throughout a time of over 60 minutes. That's how we do it, sometimes 90 minutes at the very beginning of our development. And by so doing, we guarantee that, okay, at least now these many primers which are there inside the tube are not interacting with one another. Sometimes we do have multiplex assays which have actually 2 times or 3 times that number of primers, and this is not going to happen. The second factor is the reaction mix itself and the composition of the reaction mix. So some not good configured reaction mixes can also promote this amplification problem. So that's what we do also, is to screen our primer sets with many different reaction mixes, compositions, and we always choose the one which is going to deliver us the best result. For the user, it's also important not to expose your reactions to temperatures which are not optimum for the reaction. So we always have this good laboratory practice, and we always also recommend it because the longer, for example, you leave your reactions on the bench and you go and drink some tea and come back, this is also always promoting some problems in the reaction setup. So there are many factors which we could recommend in this case to employ. I hope I could answer some bit of the question now.
Thank you. The second question is to Dr. Friedrike. The LAMP Primer Bank, is it your own collection or international collection?
No, it's not our own collection. It's from the University of Marburg, a German university, and they have made this collection of published LAMP assays on respiratory tract pathogens. Yeah, and it's for research use only. Yeah.
All right. Question number 3. This goes to both of you. Have you attempted multiplex detection for respiratory co-infections to detect bacterial and viral infections? And what is the maximum capacity of the multiplexing in LAMP?
You answer the first part, I answer the second part. I agree.
So far, we have not done multiplexing. But for the HoliCare panel, it's we plan to do multiplexing, but geometric multiplex. So that in one end reaction, only one pathogen is detected. But that you can detect from the same sample different respiratory pathogens. Yeah.
Yes, that's true. That's the setup in HoliCare, which we employ the geometric multiplexing. But I think the question is about really multiplexing here. And in fact, within the respiratory diseases, we haven't tried to multiplex viruses and bacteria, but I don't see the difficulty in that. That's always possible. The LAMP, we managed to multiplex until 4 targets and to detect them separately in other examples of other assays. But in terms of how many primer sets can be then put into one tube, these are much more than that. So in our publication, there are even 16 primer sets in one tube. So that's not going to be the problem. The problem is going to be how to find the filters, the fluorescence filter to detect 16 tubes at the same time. So these are the limits which I see is the number of detection of the signal, because in the LAMP, we don't have that flexibility which enables you to detect several targets using the same FAM filter, for example, or Sci5. It's always one target, one filter, or at least until this moment. Thanks.
Thank you. Question 4 is there any possibility for primer dimer as what sometimes happens in conventional PCR?
Yes, there is possibility of primer dimer. And that's, of course, very possible to have, especially when you have tons of primers more than what you have in the PCR. You should always expect that you would have problems with primer dimer and also the problems with false amplifications, which primer dimer usually causes. That's why it's very, very important before choosing any primer set at the very beginning is to do thorough testing of that primer set and also your amplification conditions, that's to say the reaction mix, and to do a lot of optimization before releasing that thing.
And to what extent this technique is successful with the targeted gene metagenomics using 16S RNA gene? I think this is in reference to the primer dimer.
No, I don't think this is in reference to the primer dimer, but to the specificity. And so far, the specificity testing we did for the Mycoplasma pneumoniae 16S primer set works really great. Like, for example, another pathogen that is closely related, Mycoplasma genitalium, we couldn't detect that one with the primer set that we designed in the end. So, and at the moment we are doing further specificity tests with further organisms. And so far the results are really promising. And yeah, so far we think that our primer set there is specific enough.
And the other question, question 6, questions regarding it regards the false positive. How do you decrease the amount of false positive primer to primer interactions results during LAMP assays? What would you recommend as the ideal reaction time limit, 15-30 minutes or more, as times above 30 minutes seem to increase the amount of false positives?
Yes, we don't, yes, 15 minutes, I would recommend this today as a maximum assay runtime for the LAMP.
Minimum runtime.
Maximum runtime, 30 minutes.
Yeah.
This is because of the tendency of these false positive problems. But you have to really verify and validate your assay and find out the conditions with regard of the sensitivity, specificity, and so forth for that runtime of the assay. This, of course, dependent also on, there is also a very important part in the LAMP reaction, which we didn't talk about here. These are just primer design, but it is how to optimize your assay in terms of reagents and in terms of concentration and so forth. And this in itself, a lot of questions are actually coming also to that part, because if you do that thing not correct, even if you have the best primer, then maybe in the end, your assay is not going to be the best configured one. So it's very important to consider all factors here and to do a thorough testing in the beginning, and also to find out the analytical values of your assay.
So moving on to question 7, additionally, how reliable are the results of colorimetric LAMP assays?
I would say they are reliable if again you configure your assay the correct way. The colorimetric reading of the results, as well as, let's say, the naked eye reading of the results, and also the turbidity reading of the results, these methods include a little bit of subjectivity there. And there are always these gray boundaries where you are not sure if your sample is positive or negative. That's to say they work very well with strongly positive and strongly negative results. The demarcation between the positivity and the negativity, there is always an area where that's a little bit lost, but that's a normal thing. I wouldn't classify them. No assay does give you 100% assurity, especially if you are receiving a negative result.
Question 8, according to your experience, what is the maximum applicon length with the LAMP technique?
The maximum applicon length with the LAMP technique, if I understand you well, you mean the maximum length which you can use to have a successful LAMP reaction. If that's what's meant by the question, I would say there is always a correlation between the length of the target, and with the target I mean now the distance between F2 and F3. They are F2 and B2. They are coming outer to that. Also the F3 and the B3 primer, the outer primer, and the shorter that you can have your primer target region, the more efficient you are going to have your reaction is going to be. I myself have tried previously LAMP reactions or LAMP primer design for a target of 400 bases, for example, just for the interest of science, but there will be no such a LAMP existing as a diagnostic tool, because it will lose a lot of the properties of the LAMP itself. It will be very slow.
The last question is, what precaution should be taken during designing primers for genetically heterogeneous species?
This already starts before when you decide when you look for the sequence and do the sequence analysis, the bioinformatic analysis, and then you should choose the most representative sequences that contain all the mutations, or make an alignment of that. And during the primer design, you have to use the specific primer design mode. And then I would check always that the position of the, if there's a mutation, that the position of this mutation is not at the 3' end of the F3, B3, F2, B2, and loop forward and loop backward primer, and not at the 5' end of the B1 and the F1 primer, because these are the most important parts of the primer for the affinity. And if this is a position where there can be a mutation in the sequence, then the affinity of the primer is not so well to the sequence if there is this mutation. And but if the mutation is at another part of the primer, so like in the middle or at the 5' end of the B3, for example, then it isn't a problem this primer will detect and amplify the sequence. I hope you can understand what I mean.
That was our last question. I would like to thank you, our guest speakers, for the interesting presentation, and also thank all the participants. You have been good listeners. You are welcome to join our future HoliCare webinars. One of them will be on biobanks. And please join the HoliCare Academy platform if you want to access all the recordings, as well as the YouTube channel. Dr. Mohammed and Dr. Friedrike, thank you very much. Allow me also to thank the technical team for making this happen. Thank you. See you next time.